what I actually did. But I was.
Oh, be able to have wine for a meal?
Yep, well, not for a meal, but 3 meals.
What's that? Oh, yeah, with the meal, that's what I meant. Oh, that, yeah, the preposition makes all the difference, right? Yes.
Yeah, we often think of what it is for meals, these meals.
Okay, let me get this right today. Recording, I'm recording. You're going to be recorded. Is that okay?
Yeah, that's fine. As long as I don't have to hear it.
Emily. There's 2 Emilys in here. Aw, you guys, isn't that nice? You got the boys over here, the girls over here. Just like a good Catholic school.
Father Sean, I wore my Crocs today so we could match.
Sweet. And Crocs with socks.
Crocs is, I mean, you ain't copy, you ain't copy.
Croc.
That's crop and roll.
Rock and roll. Saying if they're doing.
I said, I really want a picture of you with like our Crocs on. But I thought that'd be weird to ask. Is that weird to ask?
Sure.
Will they get you for us? Yeah.
How are you gonna get this?
Can you see our Crocs?
Yes.
Yeah, that's it.
Okay, I got a bunch.
Would you like to see?
Sure. Oh my goodness.
How did you have you came here to recruit you? Adam?
One day.
How did you have you came here to recruit?
Oh, yeah. I have one right now, but whatever.
Allison, what are you seeing right now? You see sciencey kind of stuff? Allison, can you hear me?
Yes, can you hear me?
Yep. What are you seeing on your screen?
It says dorsal ventral roots.
Awesome. Good. Oh, it's time to start. So we'll start. We'll get you in a second. Father, Son, Holy Spirit. God, grant me the serenity to accept the things I cannot change, courage to change the things I can, wisdom to know the difference. Father, Son, Holy Spirit. Amen. So without further ado, Juliana.
Okay. Hello. My name is Juliana and I'm a student in Bachelor of Research Thesis. I'm here to recruit participants for my study, You Decide Scientific Assignment. If you choose to participate, you will be randomly assigned a description of the criminal case and be asked to recommend the sentence and answer questions about the case and the defendant. Participation should take approximately 20 minutes and you must be 18 years old to participate. Participation is completely voluntary and you may withdraw your consent to participate at any time by simply closing the study from the internet browser. The data will be kept confidential and only Dr. Weiler and I will have access to information linking participants' students with their identities. However, that is not our intention. The professor is offering extra credit, and please email me at juliana.donaldson@stvincent.edu with like one L, one N for the Juliana. That's you. And the link will be sent to you within 24 hours. I'm recruiting in other classes, so please. Other people are recruiting for me, so please take note of the studying that side. Any questions?
And you'll send me a list of people who sign up?
Yeah.
So, okay. Thank you, Julianne.
Thank you, Bye.
See you. Bye. Okay. I'm still adjusting to have to bring 5,000 different things to class. You guys are used to having to bring 5,000 things to class. I'm not. Oh, I do have it. Good. All right. Nice. Thank you. Good work. There we go. All right. So there are spinal nerves extend from the spinal cord. They're conducting, sensory information and motor information. We talked about that a little bit. And then we're going to talk, let's talk just briefly about this peripheral nervous system. Peripheral nervous system, right? It serves 4 primary functions. It's receiving physical or chemical stimuli, right? Both from the external environment and from within the body. So both from the external environment and inside your body, and it detects these stimuli by means of sensory receptors.
Say that last part again, please.
They do this by means of sensory receptors. So a receptor, we're going to see this concept a lot. Now the sensory receptor simply, it's sensitive to a particular kind of stimulus. And then it basically translates that stimulus, whether it's a chemical or a physical stimulus, it's going to translate that, convert it into some sort of in this case, an electrical signal, which then is transmitted. Okay. But we are going to see this term, receptor, we're going to see this term a lot. Because neurotransmitters are going to exert their effects on neurons by interacting with not sensory receptors, but other kinds of receptors. Second thing is it's going to then basically transmit this sensory information to the central nervous system, the brain, the spinal cord, through afferent axons, right? Afferent, arriving to the central nervous system. Once the central nervous system makes a decision about, so the CNS synthesizes the information, decides, do we need to make a change here, et cetera, et cetera. If the answer is yes, then that information then travels from the central nervous system back out to the periphery. Okay. Along, so it then conveys this information along what we call the efferent, right? Efferent axons. And so once that information gets to the target tissue or organ, like the heart, so let's say the information is, let's say the plan is, you're in fight or flight situation. Heart rate needs to go up to prepare to either run or fight. Okay, ultimately, I'll draw my little heart here. This is supposed to be a neuron. This is an axon terminal. Ultimately, it's going to then take that information. The information is, let's say, increased heart rate. That's what has to happen. It's going to release a neurotransmitter here on the heart, which then helps make that happen. Okay. That's the peripheral nervous system. And this peripheral nervous system, it's made up of, for the most part, pairs of nerves. They're not all pairs. So I say 12 pairs, that's actually a lie. So I'm lying to you right here. They're not all, cranial nerves are not all pairs, but that's okay. There are 12 cranial nerves. But spinal nerves and cranial nerves. So the spinal nerves go out to basically everywhere below the neck. Spinal nerves going out to everywhere below the neck. Why are there pairs? One side goes to the right side of the body, one side goes to the left side of the body. So when I say pair, that means there's a right and a left. Cranial nerve is basically receiving sensory information from your face, from your head, and then they also can send information, motor information, out to your face and your head. And the main reason that I'm bringing all of this up is not for you to memorize all of these numbers. Well, we'll get to that on the next slide. So, the brain consists of, as we said, tens of billions of neurons. And you've got about 100,000 miles of axons in your head. 100,000 miles. So you can go around the world 4 times. You can circumnavigate the world 4 times if we laid out all of the axons in your head. Yo, that's a lot of nerves. You've got a lot of nerve, Emily. That's right. I can go around the world 4 times with my axons. Thank you. And we said we have somewhere in the neighborhood of 100 trillion synapses. So what is a synapse? Synapse. Yep.
The crossing of ions from nerve to terminus to axon.
Okay, so it's going to be a junction of different nerves.
It's like a functional unit between the nerve and its target tissue, which could be other nerve or the after organ.
So it's going to be a junction, right? So when you hear synapse, I use the word connection, right? Remember I used the word connection? So when you hear synapse, think of connection, okay? So it's going to be a connection between, in our case, 2 different nerves. So we said about 100 trillion synapses or connections in our brain. Oi! So what is a nerve? All right, this is the next packet. Hopefully you brought this. all of this information, I'm not going to quiz you, I'm not going to test you on what makes up a nerve, all right? All the different parts, I'm not going to do that. But the main thing is that these nerves that we talked about, these spinal nerves, especially the spinal nerves, it's not a single neuron, right? It's not a single nerve cell. It's a bundle of nerve cells. It's a bundle of nerve cells. So each of these pencil-looking things, those are all individual nerve cells, what we call neurons. Okay? This is important because even when we talked about some of those neuronal pathways, right? Neuronal pathway. Who give me one of the prominent neuronal pathways we've talked about so far? We've talked about several. Julie?
The mesocortical pathway.
Mesocortical pathway. Good. Yes, I haven't been able to spell for the last. Mesocortical dopamine pathway. It's not a single nerve. Or neuron, I mean. It's not a single nerve cell. It's bundles. It's bundles of these. Going to different places. Right? Because the nucleus in the brain is a center where we have many neuronal cell bodies. So this way it gives us some way of sort of comparing. So yeah, I'm not going to ask you to know all of these different layers of a nerve. That's not the point. The point is to give you an idea that when we talk about nerves, it's a lot more complicated than we think. It's a collection, it's a bundle of individual cells, and the same thing is going to happen in the brain. So if we have a neuronal pathway, it's not one nerve cell, multiple, many nerve cells. So let's talk about a neuron. So for some folks, this is going to be review. For some folks, this is going to be new. Okay, 4 basic structures of a neuron. Hit me. Soma, okay? We're going to get there. How about the one that looks like these little branchy looking things?
Dendrites.
So we're starting. Was that Allison?
Yeah, I said dendrites.
No, you got it. So the branchy things, right? These are the dendrites. And you notice this picture showing you the blue arrows coming in. That means information is coming into. These would all be, see all these branches, these ends. These would all be potential sites for a synapse or a connection with another neuron. Right? So the function of the dendrite is to receive information. All right, it says sensory receptors, that's true. We are going to be focusing on other neurons. And receiving the information can happen in the dendrite, it often does, but sometimes information comes to that nerve cell, that neuron, right at the, Julie?
The soma.
The soma, otherwise known as cell body. Okay, soma or cell body. So the soma or the cell body contains the nucleus, and it processes and integrates the information. Mainly happens in the cell body, but the cell body also receives information. So second function is to process and integrate the information that happens in the cell body. All right, what's the next structure that extends up? Emily? Yep. So the axon. The axon is going to transmit or communicate information from the cell body to the ends of the neuron by means of an electrical signal. Uses an electric signal. It's electric. Boogie, oogie, oogie. I'm gonna communicate with the axon. It's electric. Boogie, oogie, oogie. I'm not making videotapes. Thank heavens. Okay, good. So, I wouldn't be teaching that in school. So, we need to talk about your dancing during class. Okay, so, and what's the name for that electrical signal?
Stimulus.
Well, that would be when we're electrically stimulating, but the neuron itself generates an electrical signal. Verity. Yeah, remember the action potential? Action potential, right? Okay. So it's communicating information, if it needs to, from one end of the neuron to the next. So the axon is sort of the highway that connects the cell body with the, Julie?
What?
Got it.
Yep.
You're still writing. Keep writing. How you doing, John? You good?
Wonderful.
Good. All right, so the axon connects the cell body with the axon terminal. And the axon terminals are going to now, if there is a message that needs to be communicated to the next cell, it's going to transfer that information by means of a chemical signal. Chemical signal. This was a controversy. It was thought for decades. Well, the big controversy was if neurons are communicating with other neurons, there's a message that's somehow there's a signal that's being transferred from one neuron to the next. Some people fell into the, it's another action potential camp. Some people fell into the, well, it must be a chemical camp. And we now know that it's a chemical signal. And what's the name of that? What kind of? Emily? That's the neurotransmitter. That's the neurotransmitter. That's the chemical signal that gets sent. Neurotransmitter, that's a chemical signal that moves, is sent from the axon terminal to the next. In this case, we're going to be talking about another neuron. Now, anyways, so let's take a look at some of the shapes of different kind of neurons. All right. Here are some, these cells are all going to be found in your eyes. So we have different kinds of neuronal cells in your eyes. The cortical pyramidal cell, this is one of the primary cell that's found in your cortex. And you see all of these cell body's in here somewhere, but all of these structures here, these are all dendrites. These are all dendrites. And what we're going to see is that the dendrite itself, and look at this one, crazy, is a Purkinje cell in the cerebellum. These are all dendrites. Tell me that thing's not connected, huh? Like, Purkinje cells says to the cortical pyramidal cell, man, I got so many more subscribers on my site than you do. I feel so lame. You should. A lot of people, they want to talk to me, right? Okay. But what we're going to see is that on the dendrites themselves, there are these little structures. So this is a dendrite, but it's actually a lot more complex than this. Because the dendrite, it's not just as though the axon terminal connects with, like, a dendrite. No. There are smaller structures on this dendrite, which are called spines, dendritic spines. The axon terminal actually connects with those dendritic spines. So each of these has the potential to have multiple connection sites or synapse sites. Very complex. Can't even imagine what's going on there. All right. So, but the point of this is that even though these neurons have different kinds of shapes, they still have dendrites, cell bodies, axons, axon terminals. And they pretty much all function similarly, okay? So even if you see this, don't freak out. Don't freak out. It's just a very complex dendrite. Okay, so let's zone in now. We're going to zone in right here, right here. You see the red circle? And we call this, yes, we call it an axon terminal, but the more precise is, term for the structure where the axon terminal is actually interacting with the dendrite is called a terminal bouton or button. For those of you who are terminal bouton or button. B-O-U-T-O-N. So we're going to zone in here. Here we go. So that's where we just were. Here, boom, here we are. We got all kinds of stuff happening, and this looks really complicated, and it is. This is nothing compared to what we're gonna see that this thing is a lot more complex than this, probably next week. Okay, so what do we have happening here? The little boing, boing, boing. You see the little blue arrow going boing, boing, boing? That's supposed to be representing the action potential moving down the axon. We call that propagation. We're going to talk about that today. So the blue arrow is the action potential, that electrical signal being regenerated at each point down the axon. Reaches the axon terminal. Okay. And we're also going to talk about the normal model, the usual model. When we think about a synapse is action potential moves down the axon of neuron A, reaches this terminal bouton. We're going to get the big picture here. We're going to get the release of the neurotransmitter. So neurotransmitter is represented by these little green balls. And they are all inside these little plasma sacs or membrane sacs, which are called vesicles. And typically when you learn synaptic transmission, the neurotransmitter releases and it binds to the receptors on the postsynaptic membrane. The membrane of the other neuron. So here we see this right here, folks. This would be a dendrite, huh? This is a dendrite. This is a dendrite. This is maybe more a cell body, I'm not sure. But these up here would all be dendrites. And then we get some sort of effect on this second neuron, the postsynaptic neuron. That's the usual model. Okay, so all right. So it comes down. So this is an electrical signal. Notice the structure, these little purplish looking structures here. All right, by the number 2. They are labeled voltage gated calcium channel. All right. So what do we have going on here? Got a lot of terms here. Let's break it down so that way we can start. There's a lot of terminology. We need to know what we're talking about whenever I bring these terms up. So voltage gated calcium channel. All right. First thing, this is a cell. Yes? Yes. All cells in us, the outermost layer or the outermost region of cells is going to be that plasma membrane. That's what we see represented here. Actually, let's start here. That's what this is representing. This red is the plasma membrane. Things that are charged, like calcium has a charge. Ca+2, that means it's charged. Sodium has a charge. Potassium has a charge. Chloride has a charge. Those kinds of molecules, or those kind of molecules, cannot easily pass through the plasma membrane. They can't do it. They hit the plasma membrane and they bounce back off. So they need some help. And what we're going to see is that these channels, like they need a door to open, is what they need. So these channels are like little doorways that can open and close in the plasma membrane that allows for ions to move in or out. The term gated, that means sometimes the door is locked. Actually, let's cross out the word sometimes. Most of the time, the door is locked. Things can't pass through it. It's shut, it's closed. So when you see the term something is gated, that means it can open and it can close. The term before it, in this case it's voltage, this is what opens. This is what opens the lock. This is the key. So in this case, it's an electrical signal. Electrical signal opens this door. Calcium. When the door is open, or the channel, we're going to go to channel. When the channel or door is open, this, in this case it's calcium, is what can move through the door, or move through the channel. This is what can move through the channel.
Where does the calcium ions
From your diet. So typically, your body expends a tremendous amount of energy to actually maintain the conditions wherein calcium is high outside of your cells and low inside your cells. Calcium is high outside your cells and low inside.
Is it possible for anything other than calcium to go through that same channel?
This channel, no. There are some channels which allow both calcium and sodium to move through. But for the most part, they're very specific for a particular kind of ion. Sodium is another very common channel. Chloride channels are very common. And there are also potassium channels that can open and close, allowing for ions to move.
What would happen if something that wasn't what the channel was supposed to let in? Like what if something like sodium went through the
Potassium channel?
Yeah. Like what would happen to the cell?
The cell would probably be okay, but you wouldn't get any release of neurotransmitters. So that,
I just didn't know if there would be like a malfunction or something.
yeah, you wouldn't get the release of the neurotransmitters. That would be pretty bad.
Yeah.
So if you needed that, for example, to move, like to be able to move your muscles, and it didn't happen, you're not going to move. So it's a good question. Yeah. But these channels here, they're only going to allow calcium. So what happens then is once and you notice that these proteins, these structures here, and most of these are proteins, actually I think they're all proteins, they extend the whole width of the membrane. So they connect the outside with the inside. See that? They connect the outside with the inside. We're going to see that with every receptor and with receptors too. Receptors are always going to connect the outside with the inside of the cell. It's very important. Okay. Cool. So what ends up happening then? So as this action potential comes down, it activates. Activates means opens. Opens. So activate, in this case, it means opens. So if it's if it's a channel, this is an ion. So when you see the word activate in relation to an ion channel, that means that it opens. So it activates the voltage-gated calcium channel, which open, allowing calcium to enter. So it activates these calcium channels, which open, allowing calcium to enter. That's step one. Now, this axon terminus, this terminal bouton, contains these beige colored structures. These are little membranes. Contains these synaptic vesicles which store neurotransmitters. So they're there, they're being stored, they're waiting. Waiting for what? Waiting for the active potential to come back. So what happens then, okay, here's another term that we need to get a hold of here. Influx. When you see the term influx, that means that something enters the neuron or the cell. Something is entering in flux. That's as opposed to efflux. That means something is exiting the neuron. So in this case, we're talking about an influx of calcium. So calcium is coming into these neurons here, into the axon terminus, and this leads to the release of the neurotransmitter. So calcium comes in, influx of calcium, and this leads to the release of the neurotransmitter into space that's called the synaptic cleft. And the synaptic cleft is none other, it's labeled here. That's the gap between the axon terminal of neuron A and the membrane of, we're going to call this the cell body of neuron B. Okay, we'll get Emily, then we'll get John. Emily.
Oh, I just asked you to get back to the previous slide.
Oh, sure.
And Allison said she had a question. I don't know if you heard.
Yeah.
Okay.
Can you hear me?
I can hear you, Allison. What's going on?
So my question is when, so when the synaptic cleft stores the neurotransmitters.
The axon terminal does.
So the neurotransmitters are released from the synaptic cleft that then enter the synaptic vesicle, and then it encloses.
Can you ask that again? Oh, okay. Okay, so the neurotransmitter is stored in the synaptic vesicle. The
So it's exiting the axon terminal.
So what?
So the neurotransmitter is exiting the axon terminal.
That's correct.
Okay, I had it the other way around. So it's originating in the synaptic vesicle and then it's exiting through the synaptic cleft.
So what happens when the calcium comes in? There's a whole series of steps that happen. Proteins start moving around in the basically bind to, so there will be proteins here at this axon terminus. They bind these vesicles and sort of pull them. They physically pull them to the plasma membrane at the axon terminus. And then once that happens, then the neurotransmitter then sort of just gets released through a process that's called exocytosis. But anyway, so that's what happens.
Where
And
does,
when it leaves, then it moves. So once it leaves the axon terminus of this red presynaptic neuron, it then moves into this gap in between these two neurons, and that gap is called the synaptic cleft.
okay.
Oh, I didn't have a question. I was just telling you that Allison did that. I just didn't know.
Oh, okay, thanks.
You're welcome.
And I know I'm getting a little ahead of myself here, but then like the neurotransmitters, whenever we have to take it back into the axon terminal,
Yep.
they're stored into vesicles. They're not just free floating in there, but they're like put back into vesicles and to wait to be released again.
That's right. That's
Yeah. I was thinking, I couldn't remember if they were like already like pre-stored in the vesicles, like ready to like exit, or if they had to be like put back into it.
right. Yeah. So yeah, that's the more complex story. So we're going to definitely tell that story. But right now we're just trying to get to. We have an electrical signal that travels down. It reaches the axon terminal, opens these voltage-gated calcium channels. Calcium simply moves in. We're going to talk about why. Things just simply move in or move out soon. I hope by the end of today. If we don't, that's fine. Angry. And then this sets in motion a whole series of processes, which all I'm going to say is very simply, these proteins get activated, they literally drag the vesicles to the end here of the axon terminus. And then these two membranes, they sort of like mold and meld into one another, then the neurotransmitter gets released. Okay.
I have one more question. I'm sorry.
That's okay.
And I think you might have even touched base on this, but I just want to make sure I have it the right way. So the axon, so this, you have the cell body that connects to an axon, and then you have your axon terminal. What does the axon terminal just connect to another neuron?
Yeah, you connect with thousands of other neurons.
Okay, I just wanted to make sure.
Yep. So you see in the picture, do you see where there's another little red axon terminal on the left of the screen? Do you see that?
Yes.
And there's one on the right.
Mm-hmm.
So those can be coming from other neurons. Those are axon terminals, probably from other neurons.
Okay, that makes sense.
Okay. Yeah, so, well, let me just flip back here real quickly. So, if you take a look at this, this is very simple, but even here, if we were to say how many other neurons could this neuron actually form a synapse with? 1, 2, 3, 4, 5, 6, 7, 8. Right? Because it has 8 different boutons. And each of those boutons is probably going to have a synapse with a different neuron. Either a dendrite, typically, or cell body. Okay? Cool. These are good questions. Oh no, we got another new term. Dang, that's okay. So, and remember, the neurotransmitter, this is the chemical signal, right? This is the chemical signal that's used by the nerve to communicate to other neurons. So now, the little green structures here, these are our nerves, neurotransmitter, they're going to bind to ligand-gated receptors in the synapse. So, when you hear gated, Jonna, what do you think?
Door.
Door.
Closed door.
It's normally closed. And I'm using the word ligand. Another way of putting this would be chemical, all right? So when you see ligand, think chemical. And the ligands and the chemicals that we are going to be talking about will be either neurotransmitters or drugs. So the ligands that open specific receptors that we are going to talk about will either be neurotransmitters, in this case, or drugs. Sweet. Now, the neurotransmitter is going to have an interaction with this receptor on the postsynaptic neuron, neuron B. And it's going to interact with this receptor, and it's going to, again, we're going to say in this case, it activates. Which means that there are times when a neurotransmitter interacts with the receptor and it inhibits it. But right now we're talking about activation, opens. Okay, it activates the postsynaptic receptor. And it's going to have some kind of effect on this postsynaptic neuron. So after it interacts with the receptor, there's then going to be some sort of an effect on the postsynaptic cell. Okay, so that's the basics. The basics. Let's go over the basics one more time. Action potential. That's the electrical signal. Travels down the axon, reaches the terminal bouton of an axon, one of the boutons on the axon terminus. This activates voltage-gated calcium channels here at the axon terminus. Calcium comes in. That leads to the movement of these synaptic vesicles. What do they contain? Neurotransmitter. They move to the plasma membrane. The neurotransmitter gets released into this synaptic cleft. The cleft is the gap between the presynaptic neuron, neuron A, and the postsynaptic neuron, in this case neuron B. Travels across that gap. It is a small gap, very small gap. Binds to receptors, ligand-gated receptors on the postsynaptic neuron. We're going to say, at least for now, it activates them. And when it activates them, that then is going to change. It's going to exert some sort of a change on this postsynaptic neuron. Questions? John?
Is this like a reoccurring process? Like, does it constantly keep happening?
Yeah, constantly. Yep. So that's what happens once the action potential reaches an axon terminus. But the big question is, how in the world do we generate an action potential in the first place? So if you've had this, or Jen Bai, you've had this before. But here we go. We're lots of sciencey kind of stuff, lots of physiology stuff right now. We'll get back into the more conceptual stuff in a little bit. So we have to somehow, there has to be something that actually generates this electrical signal, okay? And in the electrical signal that's generated, by the way, this is a decent information from your book. It's good. Mine's better, but that's okay. Mine's more right. But anyways, it's all right. I'm moving you, Allison. There we go. Okay. So the action potential is the electric signal that is sent from the cell body to the axon terminal. And it has these properties. It's transient, very fast, like it's a millisecond long. That's 1/1000th of a second. Fast. Can you even count the 1/1000th of a second? You can't. It's regenerative. That means that if it were to be initiated here and were to be traveling this direction from the left to the right, it could then, if I generated an action potential here, I would then see one here, 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 here. It keeps getting regenerated. Remember the boing, boing, boing? That's what I mean by regeneration. And the action, the voltage, all right, this is the voltage changes. So this neuron is actually electric. It's actually electric. So what is this structure here? This is a voltmeter, and this is a recording or a reference electrode, which is on the outside of the membrane. And then you insert a recording electrode into the neuron. So one of these, the recorder is on the inside, and then the reference electrode is on the outside. Simple thing is it's when you see, for example, the region here where the tracing is black, this is called resting potential. And this is the voltage. So what's the voltage when tracing is at rest, Emily?
At resting,
Is it positive or negative?
negative.
So at rest, the voltage is -70, which means that the inside of this neuron is negative. If we were to draw that, maybe I'll do that. If we were to draw this, all right, it's oftentimes represented in this way. Here's my neuron. I simply just put some minuses on this side, and I put pluses on this side. That's all we're saying. All this graph is saying is that when this neuron is at rest, quote unquote at rest, that the inside is negative. And during an action potential, what happens? It becomes positive. During an action potential, this would actually reverse. The inside would become positive, the outside would be negative. And one of the things that we're going to have to think about is the term potential. These are membrane potentials. Potential. Why the term potential? Well, first of all, why is it called a membrane potential? Because it's a potential that exists across the membrane of the cell. That's why it's a membrane potential. But why do scientists use the term potential to talk about this?
because of the all or nothing concept?
Okay, it's about the all or nothing. There's potential. Think about there's the potential for something to happen.
The potential for stimulus?
Well, potential for stimulus, but what about in response to the stimulus?
The change in gradient,
Changing what kind of gradient?
like the positive and negative, like switches to release the electrical signal.
Okay, good. So, back up just slightly. John,
So,
what's up? What's up? Euro. I don't mean whatever. Okay, so when calcium moves in here, what do you think happens to the charge in the membrane inside the cell right here? What do you think happens to it? Become more positive or more negative?
positive,
More positive. Okay. So the membrane potential, there's the potential for ions to move in across this membrane. There is the potential for ions to move across this membrane in one direction or the other. That's what that, when you think about that, okay, the potential. And at rest, the inside is negative, the outside is positive. Okay. So during the action potential, during the action potential, that's this red region right here. During the red region, this rising region called depolarization, this is the rising phase. What's happening to the membrane potential during this phase? Becoming more negative, more positive, not changing.
becoming more positive.
Becoming more positive. A lot more positive. So, that's because ions, cation ions, which are positively charged ions, we're going to see are moving into the cell. What about this other phase, the repolarization? This is called the falling phase. What's happening to the membrane potential? Julie?
Going back down to negative.
So it's becoming more negative, right? And then we see at the very bottom, so you see the blue portion where it actually becomes more negative than the resting potential. That's called hyperpolarization. So, these are two important terms that I want to highlight here. Because this is going to come up probably next week. Depolarization. Depolarization is when the membrane potential becomes more positive. That's depolarization, when the membrane potential becomes more positive. Okay, and then I want to highlight hyperpolarization. Now, in an action potential, it's this blue, they call it after hyperpolarization, but in general, when hyperpolarization is when the membrane potential becomes more negative. Oh, I'm going to qualify. Than the resting membrane potential. And I'm just going to abbreviate resting membrane potential with RMP. And I want to qualify depolarization as when the membrane potential becomes more positive than the resting membrane potential. And this is going to become important to keep these two terms straight in your head, because there are going to be neurotransmitters that are released that can make the membrane potential more positive. That's a depolarizing event. And there are neurotransmitters, when they bind to their receptor, that make the membrane, the plasma, the blah, 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 the membrane potential more negative. That's hyperpolarization. Now, why this is important, we're going to see maybe today, probably not, but in order for the neuron to generate an action potential, the membrane potential has to become more positive, sufficiently positive, to reach what's called the threshold potential. That's important. So in order for a neuron to generate an action potential, the membrane potential, which is simply the separation of charges across the plasma membrane, at rest is negative on the inside. It has to become sufficiently more positive to reach what's called the threshold potential. And when the membrane potential of a neuron reaches threshold, boom, action potential. That's important. So if you didn't get that, get it off the video. It's really important. Julie.
So if it doesn't reach the threshold of -50, but it still like increases, it still becomes more positive, what do we call that? Like, that's say still resting potential?
Nope. We're gonna get there. That's still a depolarizing event. But it's not sufficient to generate an action potential. So in sensory, you get, if you were to, for a sensory receptor, you can stimulate the sensory receptor. But if you don't stimulate it enough to raise that sensory receptor to threshold, like say you're touching something, if you don't stimulate it sufficiently so that way you generate an action potential in that sensory neuron, you don't feel it.
That's why it's like all or none, really, because you have to reach that threshold where it's like, get that potential for that with strength, the size, it does not like matter if you don't reach that threshold when you get like nothing.
That's correct. Yep.
I think it's not a question, it's maybe an answer, like, to one, that like one dendrite can, so multiple axon terminals can connect to one dendrite. So one axon terminal doesn't have to completely cause the action potential. It could be like 7 axon terminals causing a little bit of depolarization. They together will.
We're gonna get to that.
I just hopefully might help on the understanding.
Right, so you've got multiple axon terminals releasing neurotransmitters on a neuron. Some of them are going to have a depolarizing effect. Some of them may have a hyperpolarizing effect. And then it's what's called the summation of all of that input that determines whether or not you're going to get an action potential at the next neuron. We'll get there. Okay. Questions? Let's cover this, and then we're going to, we have a quiz next week, right? Monday.
Yeah, it's online, right? Liz?
Yo.
Then yeah, Monday.
Open to me. Okay. So let's see what happens. All right. So how? Oh, I gotta move Allison again. Allison, you good out there? You chill? You
Yes.
awake?
Yep.
Okay. I can't see you now, so I'm afraid you're like I don't know, watching your cat or something like that. Anyways, okay. So let's take a look and see how the movement of ions across this plasma membrane contributes to the action potential. This concept is going to be really important because this will help us make some predictions about when I say a particular ion can move, whether that's going to potentially inhibit the neuron, postsynaptic neuron, or potentially activate it. Inhibit, so activating will be like depolarizing, making it draw, raising the membrane potential closer to threshold, and inhibiting would be this hyperpolarization, making it more negative in relation to the threshold. Okay. So, depolarization. Now, here we see concentration differences. I'm only going to talk about two major ions right now. Here's our plasma membrane. Outside of this neuron, sodium is high, potassium is low. Right? High, low. So, if I were to drop this chalk, if I were to let go of this chalk, what's going to happen to it? Is it more likely to go to the ceiling or hit the floor, John?
Wait, sorry, what did you ask again?
John, I'm just joking. Chalk. No. Let's use the crock. So if I drop the crock, if I let go of the crock, is it more likely it's going to hit the floor or go to the ceiling?
Four,
Why?
gravity.
Gravity. There is potential energy for this to move downwards, right? Oh, boom, that's what happens. So, if the plasma membrane were to all of a sudden have a bunch of channels open, and we would have one of these ions move, and they're just going to move, the door's open, and they're just going to move. When they move, they're always going to move from high to low. Just like the crock is always going to move, it's always going to fall. It's going to hit the floor. Why? Because it's going from high to low. That's the same principle. And if the plasma membrane were to become more permeable to sodium, which direction is it going to move? From outside to in or inside to out? John?
Inside out. You said high to low, right?
Sodium.
Oh, sodium. Sorry, I thought you said potassium.
I know. I switched.
It's gonna go, I just lost my train of thought.
High to low.
It's gonna go inside.
It's going to go in. We call that, is that influx or efflux?
Influx.
Good. Boom. Got it. What about potassium now?
It's gonna go outside.
Is that influx or efflux?
Efflux.
Good. Okay. Those are good things to be writing down. So, let's put on our predictometers, shall we? This part of the lecture is brought to you by Predictorology. I got my predictor on. Whatever. Okay. That was like, I needed to take 5 seconds to amuse myself. I'm done now. All right. Questions? It's a drug-free day. You better believe it. So, During the depolarization phase of this action potential, which of these ions is moving? The inside is becoming more positive. So which of these ions is probably moving? Sodium moving in, right? Because sodium has a positive charge, +1 charge. Sodium's coming in. That's what happens here, sodium in. And oftentimes, when a neurotransmitter binds to its receptor, there's going to be times when it's going to activate a sodium channel. And you need to think, oh, that means sodium is going to go into that neuron. That has the potential to activate that neuron. What about during the repolarization, when the inside is becoming more negative? So we're only talking about positively charged particles here, right? So one way that I can make the inside more negative is to pour in a bunch of negative stuff, right? Well, we're not doing that in this case. So how can I make the inside more negative?
Take some of the potassium out.
Take the potassium out. That's exactly right. So during repolarization, the membrane becomes permeable. We could say that potassium channels open, and we get a potassium efflux. I'm removing positively charged potassiums from the inside of the cell, making it more negative. And there are neurotransmitters that exert their effects on the postsynaptic neuron by ultimately opening potassium channels, which allow potassium to leave. That's a real effect that a neurotransmitter has. Okay? And then we got to talk about this. Yes?
So you said for depolarization, the sodium is going in, and then for hyperpolarization, the potassium is going out?
For re and hyper.
Okay.
In this case, in this action potential. Okay, this is a little different than what happens at a synapse. Okay, but this gives us some important principles. All right, major principle. We'll review this on Tuesday. So, quiz. Oh my gosh.
Are the quizzes timed?
Huh?
Are the quizzes timed on board?
Yes. Quizzes will be timed. Okay. You got a bunch of terms, a lot of different terms. Be familiar with those terms. We talked about brain areas, and we also talked about like the lobes of the cortex, important. I definitely highlighted specific brain pathways. You familiar with those pathways? Where they originate, where, for example, right? Substantia nigra, movement, tegmentum, right? Boom. What's the neurotransmitter? Dopamine. So if there's a pathway, know the neurotransmitter, know the name of that pathway, and where it starts. That's important. And if I gave you any, we'll keep it there for now. And then when I there were times when I said this is just for your information, like all the parts of a nerve. I talked about how the nerve is actually comprised, right? All the different layers. That's for your information. That's to give you a conceptual framework to understand that, especially when we go into the brain, we're not talking about one nerve cell. We're talking about networks of nerve cells working together, being connected to different areas. Cool. I've never taught this class before, so we'll see, all right? So it's gonna be, I will be merciful in grading.
Thank you.
See you, Alice. And do you have questions?
So the quiz, it's open for 48 hours, right?
I do believe so.
Okay. That was all the questions I
Okay,
had. Have a nice day.
On Monday, or can we do it before Monday?
I have to look.
Okay.
I don't remember. I will post them. Oh, you want to put it? You good?
Thanks, Sean.
See Julie.
Have a good weekend.
See
We don't frame
Donna.
that picture.
Oh, have a good weekend. Hey, you too.
Hey, I was gonna thank you again for letting me like sit in on the class. It's like a great refresher. Of
Okay,
information, like I really appreciate it.
you're welcome. You're welcome. And if there's anything that, if there's anything I actually, I kind of goofed on my information about auditory. Auditory doesn't go to the the primary sensory cortex.
It doesn't?
I don't think so. I think it goes to right to the, I have to look that up. I haven't seen anything stating that it goes to, of course there's I don't know. I've read the neuroscience, basically. You guys didn't use Kendall, no, you didn't. What book did you use? You used neuroscience, right?
Yes. No,
It's hearing is, did you see the really cool video about hearing, about how it works?
no, I didn't.
Of the
Yeah.
Did you check that out?
Not yet, but I will have to do that.
It's really cool how the inner ear works.
Oh, yeah. Like you just do it and everything.
Yeah.
This study was a bit about that.
So,
Yeah, there are certain things like I remember better than like others like from neuroscience class. So it's like this stuff like it's fun, but it's like the auditory stuff, like a lot of the systems. It's just like it's just a nice refresher for me to like to come in and just like remember all this stuff. With the quizzes, I think you told me before I could still take them past like test my knowledge.